That is a Swedish astronomer who succeeded his father as professor of astronomy in Uppsala in 1730, and he developed a fixed scale of temperature. 0 degrees, water freezes; at 100 degrees, water boils, and he was born back in 1701. Move forward about 100 years, and in 1853 was the death of William Beaumont. And the gastroenterologists in the audience should remember, if they don't, that name. He's an American Army surgeon who is known for his experiments related to stomach function and digestion. And basically, as a surgeon, he was asked to take care of an individual called Alexis St. Martin, who was wounded and developed essentially a big wound that was the size of the palm of his hand. And this man survived, but for the next few years, Dr. Beaumont was able to look into that stomach through a fistula and actually obtain secretions over the years. And what he did is he understood what's happening in there, identify hydrochloric acid as an important component of gastric juice, its bacteriological properties, and made a number of conclusions about how the stomach works and how motility and secretion happens, and those things are still important today. Now move 100 years further, and in 1953, Nature publishes a one-page article about DNA structure by Watson and Crick that won them the Nobel Prize in 1962 for medicine or physiology. But today is about pollution, so I'm going to bring you back to the year 79. Yes, you heard me, 79. The long-dormant Mount Vesuvius erupted in Italy, burying Roman cities of Pompeii and Herculaneum in volcanic ash. Ash was the least of their troubles. 20,000 people died that day. With that, I'll give you Dr. Roberto Bolli, who's going to introduce our speaker.
Thank you, Jesse. That was August 22nd, around 12 noon, when everybody was startled by the eruption. It's an amazing feat. So, it's my pleasure today to introduce my colleague and friend, Aruni Bhatnagar, that most of you know very well. At the outset, let me say that Aruni has been the best recruitment I've made in Louisville in 19 years. So when you recruit faculty, it's like when you buy stocks, right? You do your research, and you talk to the analysts, and they tell you all the prospects of the business, and you pick the best. You buy 10, and out of 10, 5 or 6 perform just about as you think they will. 2 or 3 will be lemons, and 2 or 3 will be just, way beyond your expectations. And Aruni is that one stock that has gone way, way beyond the expectations. It would have been like buying Microsoft in 1985, basically. He has performed incredibly well. He has built a whole research group of his own that is focused on environmental cardiology, as you will hear in a second, and diabetes and obesity. So Aruni got his Ph.D. in chemistry at the University of Kanpur in India, then came to Galveston, Texas, and was a research fellow there, and then faculty as assistant and associate professor. And in '98, I was able to snatch him and lure him to Louisville. And I have to tell you a little story how I recruited Aruni. So I was on a study section at the NIH reviewing grants, and I see this application from Dr. Aruni Bhatnagar in Galveston. And I thought it was absolutely atrocious. So I gave it a very, very low score. The application did not make it. And so Aruni was basically, out of luck. He had no money, no resources, he was despondent. And a few weeks later, I get a letter from Dr. Aruni Bhatnagar inquiring about potential opportunities in Louisville. And so I said, that's what you have to do to recruit a faculty. Just starve them to the point of total utter desperation, and then they will come begging for a job. Right. So that's how it started. But as I said, he has been incredibly successful. He was the PI on a program project grant in Louisville at the time when there were only 2. And then he built his COBRA grant, which is focused on diabetes and obesity. He's director of the Diabetes and Obesity Center. He is one of the most prolific writers, has published 170 papers, countless reviews, editorials, He's a regular speaker at national and international meetings, and serves on a variety of study sections. I lost the count. I mean, you would take 1 hour just to list all the study sections where he's a member, and he was recently appointed as a regular member of another study section. He's clearly viewed as one of the leaders in cardiovascular research in the United States. He is the father of environmental cardiology, which he will talk about in a second, whereby he discovered the influence of pollution and pollutants on cardiovascular disease, an influence that may not be as big as cholesterol or other risk factors, but it's important because it is totally preventable. So it is a very important consideration there. And then he recently focused on diabetes and obesity, which obviously are enormous problems, particularly in Kentucky. He is the deputy editor of Circulation Research, and on that, Aruni, you can say that you are on a par with Dr. Eugene Braunwald, because if you look at the masthead of the journal, there is Dr. Braunwald on one side, senior editor, Dr. Bhatnagar on the other side, deputy editor, and you are on the same line. So you can say, I am on a par with Dr. Braunwald. Seriously, has been also an incredibly helpful editor for the journal and a source of tremendous idea in the vitality and inspiration. So we are very privileged to have him here. We are thankful for his enormous contributions to the University of Louisville, and we look forward to his lecture on environmental cardiology.
Thank you, Dr. Bodhi, for that very kind introduction. I don't know if I can live up to the expectations. When I first, Dr. Bodhi came to me and said, well, you want to be a deputy editor of circulation research. I said, what am I going to get out of that? So he said, you know, when you walk on the streets, people will sort of bow to you. But unfortunately, that never happened. But I'm still trying to do the job as best as I can. So my sort of self-appointed task this morning is to talk to you about the environmental factors that contribute to heart disease. And I thought I would approach this topic in a very general sense, to give you an overview and sort of my personal opinion about why I think the environment plays an important role in heart disease. And secondly, to present some of the recent data that we have collected over the last 4 or 5 years supporting this concept, how the environment impacts heart disease and how we can measure this impact. So as you all know that cardiovascular disease is the leading cause of death in the United States, and it is a collection of different clinical outcomes. It could be stroke, changes in blood pressure, heart failure, but the coronary artery disease accounts for a majority of the heart failure or the cardiovascular deaths in this country. But it's true not only for the United States, but the rest of the world as well. And it is a leading cause of death, and it's been so since 1900, except I think in the year 1917 when the Spanish flu was the leading cause of death that year. But since then, it has sort of outpaced all other forms of mortality. It kills about 3 times as many people as all forms of cancer combined. And it is sort of creeping up not only in the developed world, but in the developing world as well. Even in middle-income countries as well as low-income countries, heart disease is the leading cause of death. And in fact, about 70% of all cardiac deaths are from low or medium-income countries. So it is the major cause of death in all human populations, regardless of geographic locations, economic status, or ethnicity. It sort of is a non-discriminatory disease. So from this, the question arises that is heart disease a sort of a system flaw in which the humans are made? Maybe there is something intrinsically about human physiology that predisposes people to heart disease. And if you live long enough, and if you're free from infectious diseases, it seems to be an inevitable fate. In fact, of people who are over 80, about 75% deaths are cardiovascular deaths. So is there a design flaw? And if there was, how would you go about finding it? So I started looking at not just the humans, but our closest relatives, and a distinguished family of our cousins who are in the wild very similar to us. We diverted from the chimpanzees about 5.2 million years ago, and we still bear a very striking resemblance to that. There is some literature about the causes of death in the great apes, both in the gorillas, the lowland western gorillas as well as chimpanzees, and they live about 15 to 20 years in the wild, but in captivity can live up to 40 years. And the cause of death is heart disease, but it's a very strange sort of heart disease, kind of a cardiomyopathy and cardiac fibrosis, which leads to congestive heart failure, and it's something that we really don't see in humans. So they have a very different type of heart disease, but certainly not the garden variety atherosclerotic heart disease or coronary heart disease that we see in humans. In fact, that's quite rare in great apes. So from this analysis, it seems like humans have no family history of coronary artery disease, and therefore there must be something in the human condition itself, rather than in our genetic origins, that predisposes all of us to heart disease. The genome for the chimpanzees was sequenced several years ago, and what was found that there's about 99% similarity between the genes in chimps and in humans. There is 1% difference, and that's a very stark difference, as you might be able to tell. Humans have a much greater cognitive ability, but the metabolic genes and the genes that are sort of contribute to heart disease in humans are the same as in chimpanzees and in gorillas. In fact, several of the sort of disease-specific alleles, such as the PPR gamma or ABC1 transporter, in the wild-type alleles are the same as in chimps as in humans, meaning that in the chimps they do not predispose them to heart disease, but they become disease-causing alleles in humans. So even with the same genes, the outcomes could be very different. And the reason for that is that we are all adapted to live in a certain environment, and an ancestral gene can only be functional in that particular environment. And if you change the environment, then you could actually have an ancestral gene become maladapted. So a gene for PPR gamma that was adapted in the environment of a chimpanzee is not adapted in a human environment. And then if you have positive selection and you have a derived allele, then you can adapt. The surprising part of that is that most of the cardiac or the genes that contribute to cardiovascular disease are actually under positive selection, particularly genes such as ApoE and angiotensin, which argues against the fact that cardiac disease is a later life phenomena and that there is no evolutionary pressure against the selection of these genes. So there may be something that drastically changes in the environment that makes these genes sort of a risk factor for cardiac disease. And this risk arises because the environment changes so rapidly, particularly the human environment, changes so rapidly that it becomes that the gene does not have the time to adapt to this new environment, causing this increased disease risk. So I will show you two examples. So one is the ApoE gene, which has different variants. This is the ancestral allele is epsilon 4, which is sort of a low frequency in humans, but this is the allele that is present in chimpanzees and in gorillas. And in them, it does not associate with increased cardiovascular risk, but in humans it is. The other interesting allele is the epsilon 3, which evolved around 100,000 years ago, which, if you recollect, is the beginning of the human civilization with the time that agriculture started. And it is believed that this mutation was because of the close proximity that humans lived with animals. And so there was an adaptation to these various forms of infections that humans were getting from animals. And so therefore, in a human environment, or the environment in which humans live, this gene is associated with a low rate of heart disease and protection against infection and head injuries. Apparently, the primitive man had frequent head injuries. And so this was a very sort of important selection factor. The same could be said for the angiotensin gene, because according to modern concepts, humans evolved in the hot savanna in Africa, and this gene was important for conserving salt. And the frequency of this allele shows a sort of latitudinal gradient. Actually, there are interesting studies showing from north of Africa all the way to north of Europe that you could see the gradation of the allele and how it varies as the humans migrated northwards. So, and it is more common in Africans than in Europeans. And the Europeans have acquired a derived allele, which is again under positive selection. And therefore, individuals who have evolved to live in a hot, arid climate are more susceptible to hypertension. So certainly it is not only an immediate environment that's of great impact to and a great determinant of a cardiovascular disease risk, but also our ancestral and evolutionary history that contributes to our disease risk. So if we are going to understand the environment, we have to first systematically evaluate what do we mean by the term, how are we going to look at the different sort of categorically differentiated forms of the environment, and how can then we tease out each of the impact of each of these components. So for that, sort of I put this together to get across the idea that there are various forms of the environment. So the most sort of ubiquitous and invariant form of the environment is a natural environment, and I'll show you some data how that impacts our disease risk. There is also the community environment and the community we live in, and the personal environment. So notice that these environments are nested within each other. Certainly before the human culture started, we were only sort of susceptible to natural environment, but we have built this oyster's shell around ourselves. So now our evolution is driven more in part by our immediate, what I call the plastic environment than the aplastic environment, that is the natural environment. And so there are several features of the natural environment that have a profound impact on our disease, such as the night and day cycle, the seasons, the sunlight, and the altitude, whereas features of the community environment also have a profound influence on our disease and our disease outcomes. Then, of course, we have a personal environment, which some people call lifestyle choices, and I'm sure the cardiologists here know how important those are in both disease risk as well as disease outcomes. So we sort of, briefly, I wanted to go over each of these components and see how they contribute to overall cardiovascular disease risk. So the most sort of primitive or the most sort of common environment is the feature of our environment is the diurnal cycle, and all life is sort of synchronized with this day and night cycle. And so is the cardiovascular function, because cardiovascular activity as well as neurohormones that regulate cardiac function change rhythmically and in synchrony between the night and day cycle. And about 13% of our genes can change in a matter of during a day. And I'm sure the cardiologists know this well, that myocardial infarctions are most frequent between the early morning hours than later in the day. And if you perturb this environment, that you could have higher rates of disease and obesity and adverse cardiovascular effects. In fact, some people suggest that this continuous lighting has had important implications on our disease, particularly in breast cancer outcomes, even in cardiovascular outcomes. There was one interesting study that we are so synchronized to environment that people who were in Japan and came to Hawaii for a holiday would actually have their heart attacks at 4 to 6 a.m. Japan time for the first 4 days of their stay in Hawaii. After that, they were synchronized to Hawaiian times. So we are synchronized to this light and day cycle, which is a very primitive feature of our environment. And any disturbance in this pattern precipitates disease. We are also attuned to the climate. Several studies have shown that cholesterol levels are particularly higher in winter and summer than in summer, and people don't know that. Perhaps it is our sort of ancestral history of hibernation and accumulating fat during the winter days than in summer. There are seasonal variations have also been observed in several major cardiovascular risk factors, which would predispose us to greater disease or greater events in winter. In fact, in England, it's been shown in winter there are 20,000 additional deaths per year. And so the same data from the United States. And some people believe that this is because in winters we lived in closed environment, and then we have more recurrent infections and therefore more cardiovascular deaths. But data from California, where there really is no winter to speak of, also shows that there is sort of a dip in the summer months and increase in winter. And we don't quite understand this sort of seasonal variation in cardiovascular mortality. Even in animal experiments, it's been shown that in times of winter it would be more susceptible or more high rates of cardiovascular disease than in summer months. So one factor that will contribute, of course, that changes in winter and summer could be sunlight, and we are also entrained very closely to sunlight. We are actually, like plants, we photosynthesize just like the plants do, and we adapted to each latitude of the of Earth, so that we have evolved to live in one specific latitude, and we move from the attitude that we are evolved to live in would cause a sort of mismatch between our photosynthetic abilities and our ability to ward off disease. So we synthesize vitamin D in northern latitudes, such as, for example, Boston. The ability to synthesize vitamin D is completely prevented from November to February. And if you look at the records, and these are old records from Irish physicians, and even up till the early 1900s, about 60 to 70% of Irish kids had some form of rickets. And so also in Northern America, in New England, rickets was very, very high. And this was eventually overcome by putting vitamin D in all milk in the United States. Unfortunately, several of the native people in the United States could not digest milk because they don't have lactase genes, so they had to resort to other things. So in any case, about 3% of our genome is regulated by vitamin D, and all cardiovascular tissues, including the heart, endothelial cells, smooth muscle cells, have receptors for vitamin D. And it's been shown that people who have low levels of vitamin D have high levels of cardiovascular disease. And there was a recent analysis a meta-analysis about 18 studies sort of involving about 57,000 people, showing that there was about 40% reduction in cardiovascular disease with about 500 international units of vitamin D intake daily. The IOM had a panel which sort of debated this for about 2 years and did not come out with a clear recommendation. The problem is that if you have excessive vitamin D, it could cause calcification in the aortic valves and could cause other problems. So we haven't really figured out whether there is a universal standard, or different people with different skin colors actually need different amounts of vitamin D. But it remains an important cardiovascular risk factor. Vitamin D also regulates blood pressure, and this was an interesting study showing that if you move away from the equator, you could actually have higher levels of blood pressure. So the prevalence of hypertension increases as the distance from the equator increases, and so we are vetted and quite embedded and rooted in the latitude that we have evolved. And certainly cardiovascular events are higher in people who have low levels of vitamin D. But not only the sort of cycles of night and day, the seasons, but also the altitude makes a huge difference. So there are nearly 150 million people who live at very high altitudes. The oldest are the Tibetans, who have lived there at least for 50,000 years, and they have adapted to the low pressure and have very low rates of heart disease. Whereas the Andean natives, they have a shorter history, have greater muscularization, particularly of the right pulmonary artery and the right ventricle, as part of what is known as kind of a mountain sickness, and therefore are more susceptible to heart disease than people with longer ancestries. But there are a lot of other cultural and geographic features that would contribute to that. The interesting study was that this was from Switzerland, and it showed that people who lived in high altitudes had much lower risk of heart disease than people who lived sort of in a near sea levels. And even when they moved up, they did not change but if they moved down, they still had a slight sort of advantage. So if you are born in a high altitude and you've lived early life in a high altitude, then you have a lower risk of heart disease. Again, we do not know the reason why. And it's not only true for heart disease, but also neurodegenerative disease, and Alzheimer's disease is much higher in people living near sea level than people living high up in the mountains. And the benefit sort of continues for an additional 10 years if you move from a higher altitude to a lower altitude. So certainly these are very important factors that contribute to our overall disease risk. But even within a particular lifetime and within a short span of time, changing in the environment could make a difference in the disease risk, which again shows it's like a natural experiment. So it's not just like there's a correlation between health and certain features of the environment, but if you change the environment, you can change the outcome. And that's sort of brought home by several studies showing that people who move to a different environment, for example, Japanese men moving from Japan to San Francisco, Indians living in the United Kingdom, or Finns migrants in Sweden, show that there could be differences in cardiovascular disease risk just for moving from one environment to another. There are also within the same community, if you don't move and you're in the same community, you could have a difference in, So you could have the same risk. Your risk will be changed by just changing the environment. So in Finland, before they were part of EU, they had these subsidies on pork and dairy products, and the heart disease rates were very high, and everybody above sort of 50 had heart disease. But when they became part of EU and changed the nutritional programs, getting fresh fruits and vegetables from the Mediterranean countries, there was a 75% decrease in mortality just over 20 years. Same thing happened in Poland, where they changed the consumption of oil, and surprisingly, or maybe not so surprisingly, in China, with the increase of Western culture and the sort of availability of fast food, there has been about a 50% increase in mortality in a very short period of time. So certainly in this time period, your genes are not going to change substantially. So changing the environment could have a drastic impact on cardiovascular disease. Not only is this the location or the particular area that you live in, but also the type of economic conditions that you are in makes a huge difference. And this was an interesting study, I think part of that from the ERIC cohort, showing that even if you have the same socioeconomic factor, which by the way is a very important risk factor, even within the same socioeconomic factor, if you live in disadvantaged neighborhoods, then you would have a higher risk of having cardiovascular disease than if you lived in a neighborhood which is cleaner and then more secure, and then you feel more firmly grounded and have a sense of belonging to the neighborhood, that you would have a much better sort of cardiovascular disease risk profile. And it also depends upon the type of people we associate. And so there have been a lot of studies showing that there is some sort of genetic association with cardiovascular disease. So even with, say, the fatty acid transport protein, there is about 5% increased risk if you carry that allele. But if you live close to a person or you have a friend who's obese, your chances of getting obese are 120%. So just being around people who have high rates of heart disease, of high rates of obesity, it could be an important risk factor. And this was actually a very brilliant reanalysis of the Framingham risk study. And the Framingham study, they showed that and this cohort was followed from 1943 up to date, and they showed that the obesity spread in networks. So obese people tend to aggregate in particular groups, and non-obese people seems to aggregate in a different group. So having an obese neighbor, or obese wife, or obese friend increases your chances of becoming obese. And same is true for heart disease, same as well as smoking. But what I really wanted to talk about was the impact of pollution on heart disease. And that's been sort of an interest in our group, and we have been working on this for some time. And it's been estimated that this is a very widespread problem. It's generally underappreciated. There has been some estimate that a large number of people, both in the developing parts of China and India, die prematurely because of exposure to air pollution. And this is not unique to India and China. In the developing years in the European history, as well, even at the time of the cavemen, where they lived in the cave and cooked meat and inhaled all the particles and smoke, there was early mortality associated with exposure to air pollution. But we have sort of excelled in changing a natural environment to the point that it has started to impair human health. Carbon dioxide levels have increased 31% since the pre-industrial era, and most of this is since the 1960s. In Europe, the pollution of the Mediterranean has increased, and the levels of ozone have increased drastically. And for every year for the last 30 years, the climate on the Earth has consistently exceeded the bounds of natural variability that we have in recorded history. Now, there is vigorous debate of what these reasons may be, but the fact is that this is a sort of a clear-cut observation that one way or the other, whether it's man-made or whether it's a natural variation, that we have to deal with this. So coupled with that, we have about, I think, 80,000 different chemicals of use in the United States, which we are constantly exposed to. We only know the toxicology of, say, about 4,000 of them. There isn't even, say, 100 kilometers of pristine environment left, whether it's in the sea or whether it's in land, in the entire planet that's not been contaminated, where you cannot find traces of PCB or mercury. in Kentucky, for instance, we have 3,000 miles of waterways, and every single mile is polluted. There is no fresh water in Kentucky left that's not been polluted. So there are many forms of pollution, but our interest has been on these ambient air particles, which are the most ubiquitous and are present in our environment here as well as elsewhere. These particles are rather small. They are sort of distributed in different types. We call them size fractionation, particle size. There is particulate matter PM. The PM10 is 10 micron particles, which are actually called thoracic particles. These are large particles that have pollution that you can see, but you can cough them out, and so they don't really have very drastic effects, although there are some effects associated with them. There is a coarse fraction, and there are fine particles, which is less than 2.5 microns, and there's ultrafine particles. And these are the ones that you really cannot see, are really pernicious because they, once inhaled, can go to the deep lung and cause consistent problems. And the initial idea was that because we are inhaling all of this, that we would have pulmonary problems, we'll have lots of lung cancer, we'll have asthma, and they do. But the surprising part was that most of the deaths associated with these were cardiovascular deaths, and that's what was startling. This sort of pollution could be derived from many sources. There could be natural sources as well as sort of man-made or anthropomorphic sources. There could be mobile sources as well as stationary sources. Analysis of the different sources show that most of it comes from automobiles, particularly diesel, but it varies between different cities. The extent of contribution of different sources is variable. And this is important because then once we know these different sources, we'll be able to then understand what is contributing to the overall risk of heart disease and how this, the impact of this could be minimized and these pollutants could be regulated. Where is the pollution most? As you can see, it's where we live most, into the Ohio Valley, where we have the maximum amount of air pollution because of both the geographic situation as well as wind currents. There is a hot spot here in Birmingham, Alabama, and there is also in Los Angeles, there is a lot of bad air. And as one of my friends remarked, Los Angeles is where good air goes to die. So we have high levels of pollution in different spots, and they sort of correspond quite well to overall mortality. So you will see that there is higher levels of mortality associated with people living in these areas. So one of the seminal studies in this area was called the Harvard 6-city studies. And what they did, they just followed people in six different cities in the United States, and in terms of their lifespan, and this is total mortality in 6 different cities, of which I showed you before the levels of pollution. And cities which had higher levels of pollution, such as Steubenville, Ohio, had higher levels of mortality than more cleaner cities such as Topeka. And this sort of correlates with kind of the extent of air pollution, particularly particulate matter and fine particles and sulfates. When the study was first published in the New England Journal, it was very hotly contested, and it has been analyzed at least 4 times by independent reviewers to assure that what the data was correct and that they were saying was actually valid and true. Now this has gone on for, I think, about another 8 years, and the data hold consistently. More remarkably, the same group of people have published that in cities in the United States which have actually decreased the level of pollution have seen an increase in life expectancy. And cities that fail to improve their air quality have seen either no change or an increase in cardiovascular or total mortality because of air pollution. This is not a very large effect, but still, in terms of overall population, it is very widespread. So the next thing was to identify what the sort of causes of death were, and as I mentioned, most of the deaths associated were because of cardiovascular disease. So this is analysis of the cancer cohort looking at the relative risk of death, and these are different forms of death. The dot or the magnitude of the dot sort of indicates the amounts of death, and most of those deaths are associated with cardiovascular disease. As you can see, the respiratory disease do not have much of an significance, and at least some of them were actually maybe negatively correlated. But that may be because the people were dying of cardiovascular disease, so we do not know what the exact meaning of these data are. But it clearly indicates that 70% of the cardiovascular death, of the total mortality due to air pollution is because of cardiovascular deaths. And that was very startling, and that's why we think that we need to better understand how does air pollution impact cardiovascular function and cardiovascular disease. So I showed you the map before of the United States showing that there were areas in which there were high levels of pollution, and the deaths are associated wherever, or the excessive deaths are associated in areas which have high levels of air pollution. And it's true not only for cardiovascular disease, but for diabetes as well. And there was a recent analysis showing that areas which have high levels of air pollution have higher prevalence of diabetes. So not only air pollution can make things worse, but maybe it could contribute to both to the incidence of cardiovascular disease and diabetes. So how does air pollution affect heart disease? Well, we've been scratching our heads for the last 10 years trying to figure out how this is. And this is a symptom of our confusion. When you really don't understand what's going on, you make all these arrows and say everything is going on. So there is some autonomic dysfunction, there is increased thrombosis, increased acute events, there is increased chronic injury, which sort of is a confession that we do not understand what the underlying mechanism is. So what we did was to then look at some of the very elemental effects of air pollution. So we said we would look at the effects of air pollution on stem cells. And the reason for that is that these are very primitive early cells, and we wanted to look at some of the most sensitive changes upon exposure to air pollution. And so one of the stem cells that we started looking at was called the endothelial progenitor cells, and it's been called many names, and I will switch names as well, but this is the most commonly used term. And these cells, people have shown that they reside in the bone marrow, and they can home to tissue injury and then promote tissue revascularization, promote injury, and if you implant endothelial progenitor cells, and this is a mouse experiment, then you can increase angiogenesis. So these have some sort of a reparative role in regenerating cardiovascular tissues. We're not exactly sure how, but at least they are being shown to be effective. So we wanted to measure these levels in humans. And for that, we went to Utah. And I don't know if you've been to Utah. This is Salt Lake City in Utah. And the city sits on the basin of these mountains around. So what happens in the city is that you would have an inversion. And because of the inversion, there is a very pronounced increase in air pollution. The PM levels go from like 5 or 10 to 100 within a day. And then they stay there for a few days, and then it suddenly resolves. So this is like a natural variation. So you could, first there is no pollution, then a huge amounts of pollution, and then there is no pollution after that at all. So and this is shown over here. This is shown over here where we see that the spike of pollution lasts a few days and then comes back and goes up again like that. So then what we did is we took blood from there was a soccer team that we recruited. We took blood from the soccer team members, and before the inversion, because we can predict when the inversion was going to come, so we took blood before the inversion, during the inversion, and after the inversion, so that we could show a sort of a dose response to that. And what we found was that as the levels of PM increased, there was a decrease in endothelial progenitor cells. And so that would mean, and this was reversible, so you would see that whenever the air pollution would spike, you would see a suppression of these cells, and when the air pollution would come back, the levels would recover. But this was only in humans, and there could be other factors contributing to it. Maybe on the days there were pollution, these people did not go out or exercise, or there would be some other confounders. So we wanted to replicate in the lab. And so what we did here is, this is on the 7th floor of the MDR building. We made an inhalation facility in which, in this rather complicated way, we sort of collect and concentrate particles from downtown Louisville and expose mice to these concentrated particles. We can analyze them as well as do a dose response curve. And so what we found that when we exposed mice to the polluted air from downtown Louisville, that we could get a suppression of these flick scar, or what we call the endothelial progenitor cells. So sort of validating the human data that what we see in humans is what we also see in mice, that exposure to particulate air pollution can suppress your stem cells. Now the stem cells have a very complex biology. As I mentioned, they reside in the bone marrow, where they are then liberated in response to VEGF, it binds to VEGF receptor, stimulates a signaling pathway, and that releases nitric oxide that removes these cells from the stromal niche and into the circulation by S-KIT ligand. So what we did was then actually treated these mice with VEGF and then try to liberate them and see whether the liberation was affected. And what we found was that upon treatment, that there was actually a decrease, showing that VEGF-induced release was prevented by exposure to particulate air pollution. But when we treated these animals with the stem cell factor, we could then release these cells out of the niche. So it means that there is some sort of a defect within the signaling pathway, somewhere between the VEGF signaling and the release of these cells by S-KIT, that is preventing them from coming into circulation, so that there may be a specific loci which is sensitive to exposure to air pollution, and we are still in the process of trying to understand what the specific mechanism may be and what the contribution of these cells may be to overall effects of particulate air pollution. But then we wanted to go back and check how these cells, or these endothelial progenitor cells, would reflect injury and cardiovascular disease risk. And although there have been several studies, we have not been able to get a handle on what type of populations are affected due to cardiovascular disease. And so for that, we started about 4 years ago what we call the Louisville Healthy Heart Study. And in this study, we recruited over 300 participants from various parts of Louisville. And these are from our cardiology clinic, and then looked at the levels of the endothelial progenitor cells in each one of them, and some markers of exposure, so that we could get a new understanding of how exposure to particulate air pollution or other forms of heart disease could contribute to cardiovascular disease risk. This is sort of demographics of our population. To date, I think we have about almost 300 now. So we had about 250 that we have data on EPCs, but we have about 300 or more participants. The interesting thing was also that we know actually the addresses of all of these participants, so we can calculate sort of local effects that might have on these people. And then we sort of characterize the whole range of progenitor cells in the blood. And you don't have to go through all this detail, but I'm just trying to show this to sort of make the point that we could separate several different populations of stem cells and then look at their biological nature and then understand how each one of these populations would respond to cardiovascular disease risk. And this is one example. So this is the Framingham risk score of this entire cohort of each individual, and we found correlations with specific populations of these vascular progenitor cells, or we call them endothelial progenitor cells. But regardless, there was a strong correlation between sort of suppression of these populations in humans as their Framingham risk score increased. And interestingly, the marker here is AC133, which is a marker of an early progenitor cell. So somewhere in this maturation pathway of the progenitor cells, that there resides a susceptibility to this cardiovascular disease risk, and it's reflective of that. And even with some well-known cardiovascular risk factors, such as the C-reactive protein, that we can find that there is an association with specific populations of these VPCs. So indicating that we could actually sort of quantify the risk by just looking at these populations. We are in the process of trying to understand if we can actually improve upon the risk estimates by putting this into the Framingham equations and see whether we get additional information, not only about the overall disease outcomes, but also about the underlying pathology in terms of understanding the contribution of thrombosis and inflammation and exposures to heart disease. But going back to our environmental objectives. So as I mentioned, we have addresses for each of these people, and we sort of could figure out how far these people are from roadways. We calculated roadway distances of the residences. We actually went into, at least for a select population, sort of went in to find out how long these people have resided in these places, and what is the traffic density around their homes. So what we found that people who were living close to the major roadways had somewhat higher levels of these EPCs than people who were living sort of further away from the roadways, and we can sort of find that in a couple of populations. And this may seem at first paradoxical because disease risk associated with suppressed levels of these cells, but we found an increased levels, and we think that is because whenever there is ongoing cardiovascular injury, there is a transient increase in the cells that is required for repair. So we think that we found and sort of an interesting feature of the environmental injury, which is here manifests as the slight increase in these progenitor cell levels indicative of some ongoing cardiovascular injury. And we're trying to now understand if we can quantify personal exposures and what particular sort of components in this exposure because of living next to major roadways is sort of contributing to this risk. There have been some, in the time that we were doing this study, there are a couple of studies come out showing that people after a heart attack, so people who are in heart failure, actually do much worse if they live next to a major roadways than if they do live further away. So this is consistent with the general idea that constant exposure to ambient air pollution could enhance your cardiovascular disease risk. So to summarize, what we found so far was that exposure to environmental pollutants such as acrid and particulate matter can suppress circulating levels of these stem cells, and that there is the exposures prevent the mobilization of these cells by interfering with specific signaling mechanisms, and that we could actually pinpoint the specific limb of the signaling cascade that might be sensitive to air pollution, and that this could not only be used to estimate the risk of exposure, but also to gain some fundamental insights in the pathophysiology of particulate air pollution and how it affects cardiovascular disease. So, to sum, I would like to come back to the point I was trying to make in the very beginning, that part of the cardiovascular disease is imparted from the environment. I discussed some parts about how air pollution could do that, but there are other important features as well. In fact, if the other features, for example, lifestyle modification and living in healthy environments, if you take all that into account, it could be estimated about 82% of the risk could be mitigated by avoiding high-risk activities or adhering to lifestyle guidelines. And this has been repeatedly shown, and to some astonishing degrees in different cohorts, suggesting that the environment is an important component of cardiovascular disease, and in a sense, heart disease is an environmentally derived disease. And the same could be said for both type 2, also for type 2 diabetes, which largely could be avoided by adhering to low-risk life factors. So what happens is that we have wrong choices that we make, the wrong lifestyle choices that we make. They keep on accruing over our lifetime, although we do not see them, but they keep accruing into ourselves. For every wrong choice that we make, for every exposure in the environment we have, and that accumulates and sort of precipitates one day to disastrous effects, like the story of Dorian Gray. So what this tells us, though, is where do we think the disease should reside? Our current understanding of the disease is that it is with the individual is making the wrong choices, and that there is this proximal cause of disease that needs to be urgently and vigorously treated. But it may not be that. It may be that the disease actually originates from the environment, and these distal causes, whether they be evolutionary causes or whether they be causes in our environment, a natural community or personal environment. So the treatment should be directed to the cause which is distal to the individual. The individual is actually, in the end, the victim of this. And so if we divert our attention to a better understanding of these extraneous influences that drive cardiovascular disease, that we would tend to gain more, at least in terms of preventive strategies, than if we keep on focusing just on the individual itself. The new thinking is that for long and many, many years, we have been talking about risk factors, and I think that is I agree with some other people, but I think that's a very morbid way of looking at cardiovascular disease. So rather than constantly talking about risk factors, we should be talking in the opposite way and talking about health factors, not sort of passively preventing disease, but actively promoting health. And that would have a greater gain than trying to sort of stop every seep through the dam that we can see, but rather to build a stronger dam to begin with. And what I was trying to accomplish, or trying to show this evolutionary history, that cardiovascular disease is not only governed by environment, but it is very sort of tightly regulated by the sort of the environment, the people that we live with. And so healthy adults, healthy children make healthy seniors. And so if we sort of understand this and begin health promotion early, that we could have much greater success in our attempts to prevent cardiovascular disease and overcome some of the limitations that we have currently trying to treat everybody in the very end. And we know for a fact that we're losing the battle because it continues to be the #1 cause of death around the world. So not only do we have to understand the molecular and cellular mechanisms and what networks operate, how these networks actually interact with each other, but also we have to address the disease networks, organ systems, cardiovascular tissues interacting with, say, hepatic tissues, how the pulmonary inhalation and pulmonary injury affects cardiovascular injury, and then finally how social networks sort of seem to influence the disease state. So this is a rather complicated, horrendous task, but we have to actually look at disease in several different planes to be able to understand this systematically. And so before I end, I just want to acknowledge several people who've been involved with these projects and related projects. Of course, people in the lab who are involved in environmental research. Jan has been a real source of support and help. He's the director of our inhalation facility and has been sort of spearheading our environmental research. Tim, Petra, and young men, Jason, Laura, and other students have been participating in this. Dr. Pope is our collaborator in Salt Lake City and has sort of worked with us on the analysis of the stem cells there. And then we have the Louisville Healthy Heart Study investigators sort of who contributed to the study, which is still ongoing. We have yet to publish a manuscript out of this study, but we are putting them together, and hopefully we'll have some interesting data to publish soon. And of course, we cannot sort of overlook the grant support. We've been supported generously by the EPA, which helped us build this inhalation facility, by the Department of Defense, by NIEHS, and Heart, Lung, and Blood Institute. And I thank you all for your attention.
Thank you so much. This was very interesting. I had a couple of questions. The first one is one that I think is in the minds of many members of the audience. You showed us a lot of information about diurnal variation, distance from the equator, distance from roads, temperature, vitamin D, that sort of thing. Based on all this knowledge that you have of where people are healthier or not, where should we move to?
The challenge, when humans first evolved in Africa, one of the problems was they quickly spread all over the world. So we have adapted to our environment. We are evolved in a particular environment, but we have the capacity to change the environment, right? And that's the essence of human civilization. So it is not where we should move to, but how we can adapt the environment to our needs. And that adaptation has to be different for the different individual depending upon the evolutionary history. So, of course, we should avoid living in polluted areas, not next to the roads and next to factories. You know, we should probably all live in high places, in alpine forests. But we cannot all do that. So we have to figure out what are the factors that contribute to the health-promoting effects of those areas and then incorporate that into our immediate surroundings.
Very good. Yes, sir. Let's start here and we'll move upward. Mike.
This was smashing. Thank you so much. A lot of the information you presented, I think, has very important policy implications, and I'm wondering if, I know a lot of this is well published, but have you or any of your colleagues been in touch with people at what remains of the Environmental Protection Agency or the Office of the Surgeon General, and are they kind of attuned to this, and are there policy recommendations coming out of this?
Right. So
Just restate the.
So the question was were there any policy recommendations that have come out of all of this? So yes, so I have been to several of the committees, both the Environmental Protection Agency, but also by the Institute of Medicine, and trying to study the effect of environment on cardiovascular disease. The EPA has strict guidelines that they set out different rules and limits that each city can have, and if you violate that, you get penalties and all this carbon tax. I'm sure you've heard all of that. But even beyond that, for example, one of the IOM panels that I was on was about these veterans returning back from Iraq and Afghanistan, saying that exposure to high pollution there caused them long-term health problems. And was that true? And we spent about a year and a half debating whether there was any evidence to support this claim, because if we did, then the VA would have to compensate all the veterans that came back from Iraq and Afghanistan. Of course, we decided that there wasn't enough information, that we cannot really pinpoint that, because we do not have specific effects that we could follow, either in the features of the disease or specific constituents of the pollutants that we can identify, say, aha, this is the reason why you had your MI. So we don't have that definite information, but we came out with the conclusion that we cannot say anything. So yes, we could make policy, but we really need to understand why we're going to make that policy. We have set limits for exposures, but beyond that, it's still an ongoing debate.
Yes, sir.
Aruni, wonderful talk, and you taught me something I did not know, that obesity is contagious, right? My question is, so you focus on EPCs, obviously that's an important consequence of pollution, but I think it's kind of a slow, long-term effect. But we know pollution can cause heart disease within minutes or hours, so there must be some immediate mechanism other than the impairment of stem cell released by the bone marrow, right?
So the question is that EPC levels are long-term changes, whereas heart attacks could be triggered by air pollution acutely. And you're right, I want to actually highlight the data which shows, this is data from Boston, showing that for every spike in air pollution, 6 hours later there's a spike in cardiac deaths. And there is one spike after 6 hours, another spike after 24 hours. So there are exact changes. There's actually evidence that EPCs could change in a matter of days, but we don't know how they can contribute to an immediate event. The underlying mechanism there may be thrombosis, that there is change in the thrombotic capacity within a few hours. And we know this very well with tobacco smoke, that with an exposure to tobacco smoke within a few hours, you could see that there is increase in thromboxane and there is increase in prostaglandins. And so there may be a thrombotic trigger that causes this acute precipitation of cardiovascular events upon exposure to particulate air pollution.
Dr. Winters,
so
that was your question.
Paramount connection between insulin resistance and cardiovascular disease, and between inflammation and insulin resistance. So my question is there some connection between the particulates, the inflammation that they produce in the lung, and some transient insulin resistance that you might have been able to see in the Salt Lake City study, for example?
That's right. So the question is whether there was a sort of an acute association between insulin resistance and exposure to PM. And that's actually a very good point, and we are working on that. We do know that when you expose, and I didn't present the data, I published at least 3 papers showing that when you expose animals to PM, that you can increase insulin resistance chronically. We haven't seen whether that could do it acutely, but if there was an acute change in insulin resistance, that might explain the changes in EPC levels as well as changes in coagulation pathways. So we need to be sensitive to that, and we are going to measure those.
Dr. Roberts?
It's really impressive with how much progress the Finns were able to make by eating Mediterranean food. 75% is a really big risk reduction number, and I wonder if the homogeneity of the Finns and their bottleneck with the 500 individuals at one time, if that homogeneity makes them have a different genetics and ApoE or something. Could you get anything out of looking for their genetics to see how they were able to take advantage of the Italian diet so much more than what seems to happen in the rest of us? Dine out Italian restaurants.
Dr. Roberts seems to have been aggressively investing in the Mediterranean diet, and he wants to get the benefit of the Finns.
The underlying. So the question is, if there is a sort of a genetic component to getting a full advantage of a dietary modification, to put it that way. So, first of all, I'm not sure. That I don't think anybody knows for a fact. But I could just speculate that the diet and, as everything that I've been trying to say this morning, is that nothing in the environment acts independently. It depends upon the genetic background. So maybe Mediterranean diet affects only people with that particular genotype and may not affect other people with a different genotype. Fortunately, humans are very, very similar. At least in terms of the major genes, we are very, very similar to each other. And that's maybe, again, going back to the evolution. At one point of human history, all the other humans were killed. There were only like 100,000 humans left. And everybody is descended from them. So our genetic makeup is very similar, regardless of what people may think that we are very different. We are not. And so maybe these major influences may not change, but there may be subtle influences that would determine the outcomes of any dietary intervention.
Questions?
Thank you, Aruni, for a fantastic talk. Please comment briefly on the ozone layer status and where, and cardiovascular disease.
So the question is, what's the relationship to the depletion of the ozone and the cardiovascular disease risk? As far as I know, there hasn't been much information about what the impact of the ozone depletion would be on heart disease. Of course, there's information about skin cancer, and we know that there increases skin cancer. But to have an ozone layer that would sort of permit UV light to come in and cause cardiovascular disease or modify cardiovascular disease, I'm not aware.
Last question.
Andrew.
So to follow up on Rita's question, so why is the national debate about pollution and global warming when the evidence seems to be so clear here that about pollution and mortality and cardiovascular risk?
So the question is, why are we debating an issue that is a foregone conclusion? That's a good point. First of all, this is in one of the committees of the EPA, and we said that we see this high levels of air pollution, and then we see increased deaths. We could just have a policy to prevent this association, decrease pollution. But the basic nature of science is to get at the causation of things. So if you do not know the cause, it is not science. So if you do not know why air pollution is causing deaths, we cannot have a scientifically informed policy. So that's the reason for going into all these mechanisms. We know air pollution is bad, so why spend my others' lives in trying to understand what the mechanism is and what the things are? So their question was that the critics of the EPA and other people are harsh enough that would only accept scientific evidence which can only be validated if you demonstrate causation. So we came back here, spending a life trying to demonstrate causation. And the second point is that we cannot just say blanket, just let's cut down all pollution. That would not be right. We have to find out which particular component of that pollution is causing the problem, and then we can regulate that particular component. For example, these particles contains cadmium, nickel, zinc, elemental carbon, and so on. And if we can find out which of these is contributing to air pollution, to heart disease, then we can regulate that specific source and that specific component.
That statement explains why we can't ban smoking or guns. Thank you. Outstanding. Very good. Very, very good.